This region at the foot of the Himalayas has very fertile soil. People here depend on agriculture, and the market stalls are filled with locally grown food. Researchers at the Institute of Himalayan Bioresource Technology in Paramvir are trying to ensure that it stays that way. In collaboration with German scientists, they're working on improving the resistance of agricultural plants.
Collecting all the information.
Such as what kind of technical devices?
Each of the partners in the project has something to contribute.
Lots of fermenter technologies. That's the exciting part of that project, that you have got your knowledge which can be complemented to ours, so this fits really nicely. And the other aspect is, of course, that we are very keen on actively contributing to the improvement of our crops for the future.
In the Himalayas, there are many plants with properties that could be advantageous to other plants. The plants that grow at high altitudes are very hardy, withstanding both cold and dryness. The researchers have identified the genes in this cinquefoil that make it highly resistant to dry conditions. The genes are going to be inserted into a plant named Arabidopsis thaliana. This organism was the first plant to have its genome completely sequenced. Here it's being dipped into a mixture containing the drought-resistant genes from the cinquefoil, along with special bacteria to help the transfer. The plant can take up the foreign DNA within minutes, but whether the transfer has worked will only be seen in its offspring. That makes the procedure far less simple than it looks.
Once you analyze the transcriptome, you get thousands and thousands of the gene which are getting expressed during cold conditions or under drought situations. Now we are looking for some novel genes, and when it comes the question of novel genes, then it comes the question of evaluation, the role of those genes.
The scientists use microscope images to identify the new genetic variants. Here they're comparing a normal, unaltered plant, grown under dry conditions, with one of the genetically modified or transgenic plants.
The vessels, they are not so well formed in the wild type, whereas in the transgenics, there's a change in the structure and stability of the vessels, the xylem. And where we are showing that this structural stability basically is because of lignin synthesis, which is higher.
The new genes have established themselves after 4 generations. The researchers conclude that the genetically modified plants can survive drought better than normal Arabidopsis specimens. But they want to know exactly what is happening inside the plant.
So right now what we do, we harvest the plant at regular intervals, and then we have to measure growth phenomena. But what I understand that Jülich, they have the system of what they call it phenotyping system.
At the renowned research center Jülich, a special facility has been set up to do the phenotyping, that is, measuring the physical characteristics of the plant. The ultraviolet light helps determine how fit a plant is.
Here we can make daily measurements of how these plants are growing and how their photosynthesis yield is changing. It gives us an opportunity to monitor in detail whether some genes have conferred drought tolerance.
Only a few institutions in the world have these kinds of measuring devices and test stands, like the stereo camera box. This is cutting-edge plant research. And there's no problem getting enough samples to study. The researchers have a machine to help with the sowing. That enables them to experiment with the Arabidopsis at every stage in its growth cycle. And it's not the only plant to get the genes from the Himalayas.
Parallel to the Arabidopsis plants, which are just model plants for us, we intend to transform several crop plants. We already have 2 plant breeders who will be cooperating with us as partners.
One of those partners, located in western India, is the one who thought up the joint project. Anup Karwa did his doctoral degree at Jülich. His family runs a breeding company in India. He's hoping to apply the results of the research to cotton plants, although they're not as easy to work with as Arabidopsis.
Arabidopsis can be propagated within a month, and it's easy to transform. It's a very small plant, and comparatively cotton is a very large plant, very difficult genome, where the gene integration into the cotton genome becomes very challenging.
The company is equipped with the latest technology. And if the plan works out, the investment will be repaid many times over. Cotton is a major cash crop in India, but the plants require a great deal of water. That's why Karwa is hoping to use the new variety with the Himalaya gene. The plant would be able to withstand dry periods and require less irrigation. This would be a tremendous boon to India's cotton farmers.
I'm very hopeful, I'm very confident that we will succeed, and we will have a very good technology which will be unique in terms of giving a stabilized yield to plants, and we will combine it with the, again, very high-tech molecular tools that would help plants not to use this gene all the time, but only in the prevailing stress conditions.
And if that attempt works, the German-Indian research team is hoping to breed other agricultural plants that are adapted to cold and dry conditions.